Yeah, good evening, all attendees. A warm welcome to all the attendees on the behalf of IHERA, BSV, and our esteemed speakers and panelists. So, as we all know, IHERA is doing a great job in sharing the knowledge and experience in the field of embryology. It has been founded by Dr. Ved Prakash with the support of Dr. Sanjay Shukla, Dr. Charujat Joshi, and these are all the leading names from the embryology. So we look forward to learn from all the esteemed speakers and panelists. And theme for the today's webinar is the journey of sperm from production to fertilization. So here I would like to welcome. So we have esteemed co-conveners today, starting with Dr. Nidhi Singh. So she is currently working as clinical embryologist at Oasis Fertility, Bangalore, and she has vast experience in the field of clinical embryology. So we look forward to hearing from her side. Next, we have Dr. Miss Yoshita Tanwar, MSc in Biotechnology, currently working as clinical embryologist at Oasis Fertility, Bangalore again. And again, we can see her credentials, certifications. So we look forward to a good learning from her side also. Next, we have Dr. Nancy Sharma from Department of Reproductive Medicine, Jindal IVF, Asant Memorial Nursing Home. So she is also having a vast experience and has done many sessions, almost 9 years of experience in the ERT. So we welcome her from the Jindal IVF. And finally, nonetheless, we have Dr. Ananta Lakshmi. She is a senior clinical embryologist from Janani, Trichy Fertility Center. And again, seeing at the credentials, we look forward to have the good learning from her side. So I would like to welcome all the co-conveners, and I would like to hand over to Dr. B. Ananta Lakshmi for carrying forward this particular session. Thank you.
Thank you for the introduction, Dr. Sandeep. Good evening, everybody. I welcome the ladies and gentlemen for joining this webinar. It is my pleasure to welcome you all for the webinar on Journey of Sperm, that is from production until fertilization. As we dwell into the intricacies of human reproduction, we embark on a voyage through the marvels of life itself. In the realm of biology, few processes are as fascinating and essential to life as fertilization. At the heart of this intricate dance of biology lies the journey of sperm, a tiny yet mighty protagonist in the creation of new life. Today, we gather to unravel the mysteries of this journey from its humble beginnings to its ultimate destination, that is, the union with the egg. Throughout our webinar, we will explore the biological, biochemical, physiological, and environmental factors that shape the journey and shedding light on the wonders of human reproduction and the miracle of life itself. So, without any further ado, let us embark on this enlightening presentation, the journey of sperm from production to fertilization. Now, I would like to welcome our coordinators of IHERA, Dr. Ved Prakash, sir. Can I have his CV?
Yeah, hope it's visible.
Thank you. Yeah. Dr. Ved Prakash is a senior consultant clinical embryologist and scientific director in Sperm Life Advanced Andrology Lab, Delhi. He has worked as a regional head embryology of North Region, Nova South End IVF. I would like to introduce our next coordinator, Dr. Sanjay Shukla, sir. Can I have his CV? He is also a senior consultant clinical embryologist and adjunct professor clinical embryology, the Department of Reproductive Medicine from Mahatma Gandhi University of Medical Science. That is also having a vast experience in this field. Coming to the next coordinator, Dr. Charujat Joshi, sir. He is a medical director of Genes India ART Bank, Indore. And I welcome you, sir. And now I call upon Dr. Charujat Joshi, sir, to say a few words and introduce the meeting.
Yeah, I want to say one more thing. Dr. Charudas Joshi recently appointed as a chairperson embryology in ISAP. Okay.
Okay,
So
thank you.
Big congratulations. And
Thank you very much. So without taking much time, it is my privilege to introduce you about IHERA. In IHERA, we always have a motto in mind that whatever is happening in the field of infertility should be shared with the knowledge. best possible outcome out of it. So on this evening, I would like to invite all of you for the journey of sperm. On the sperm as in itself, it is very important part which has to play a very important role. So let's understand from our speakers about the production of sperms, then how it develops, how the fertilization happens, and then so on. Still, the question remains whether the fertilization will happen with this sperm or not and all. So let's try to find out the answers to this. And with this, I would like to announce one more event here, which is an privilege for IHERA to present you Micro Embed, a first physical conference of our IHERA to be conducted in Gurgaon, Leela Ambience Hotel on 8th and 9th of June. So I take this opportunity to invite all of you to be there for that fantastic scientific event. So with this, I hand over to the conveners to further proceed with the session. Thank you.
Due to unavoidable reasons, instead of Dr. Nidhi, Dr. Nancy Sharma will be presenting the first topic, which is about the spermatogenesis, that is the production of sperm, steps involved in that.
So good evening, everyone. As Dr. Ananta Lakshmi said, that due to unavoidable circumstances, Dr. Nidhi could not take the session. So I'll be taking over the talk for today, and we'll try to do justice to the talk. So we are going to discuss about spermatogenesis in detail today. So what is spermatogenesis? So what is spermatogenesis? It's basically the production of mature male gametes, which are known as sperm. So today we'll be taking care of all the stages of program, how it is done, and what are the things which are involved inside in it. So as I said, that briefly what happens, approximately 200 to 400 million sperms are produced on the daily basis in testes, and then these immature sperms, they undergoes a process of maturation and then attain motility further in the epididymis. So there are a few main functions of testes, which is one is to produce the male gametes and the testosterone. The outer layer of this outer layer of testes is called tunica vegetalis, and the inner layer is called tunica albuginea. This tunica albuginea is further having septas, these kind of septas, and these septas are having further divided into lobules. These lobules have this coiled tube-like structure inside it, which is called seminiferous tubule. Seminiferous tubule is actually the site of spermatogenesis, where spermatogenesis happen. So spermatogenesis is also a compartmentalized process, which is like divided into intratubular and tubular. First into the intratubular and tubular compartments. The intratubular compartment is interstitial space, and the tubular compartment is the seminiferous epithelium. So which is structurally partitioned by the Sertoli cells into the basal and the adluminal compartments. Here is the basal compartment where few events happens, and then we have the adluminal compartment where other events happen. Other events of spermatogenesis happen. So as I already said, that spermatogenesis is also a compartmentalized process. The occultation junctions between Sertoli cells form two compartments that shows different stages and phases of the spermatogenesis inside it. So the spermatogenesis is further divided into different phases. These phases are spermatocytogenesis and spermiogenesis. In spermatocytogenesis, there is proliferation and then reductional division. Proliferation is basically the mitotic expansion, and reductional division is the meiosis. So what happened in spermatocytogenesis? One germ cell, it divides into the four precursors gametes and form spermatids. These spermatids are immature sperms. And then we have spermiogenesis, in which there is morphological transformations and differentiations. Basically, there is no cell division during spermiogenesis, but these immature sperms, they undergo a process of maturation and differentiation. So as I said, as I already told you, that this is how it happens. First, there is spermatocytogenesis, and then there is spermiogenesis. These germ layer lines in the basement here, the basal lamina, they have the precursor germ cells for it, and they are in contact with the Sertoli cells. Here, they are in contact with the Sertoli cells, which extends from the epithelial into the lumen to the tubules, and then at puberty, this spermatogonium, this starts to proliferate by mitosis, followed by the meiosis. And now we'll see the series of cell division which will occur in the first phase of spermatocytogenesis. So what happens? There is a germ cell line, there's PGC, and that PGC, they first renew themselves. There is self-renewal of stem cell pool, and there are two different populations of it. With first one, which goes into the self-renewal, and the another one which is committed to the differentiation. So one category is auto-reproduce to maintain population, and one category is committed to differentiation. So this is how it happens. There is spermatogonium A dark type cells, which are further divided by mitosis into spermatogonium dark and the pale type ones. The pale type ones are active to meiosis, and then they further divide into the type B spermatogonium. These type B spermatogonium are actually they are actually supposed to go through the primary spermatocytes to secondary spermatocyte and further to the spermatids. And one is only to maintain the population. So here we have a diploid number of set, which is eventually followed by the meiosis, is going to be the reduced number of set to the haploid. So in A type of spermatogonium, there is no active mitosis, and in type A pale, there is active mitosis with multiple divisions, from which we get type B spermatogonium. These type B spermatogonium, they further have primary spermatocytes. So this is the detailed structure for that. A type A, it replicates the DNA and then divides by mitosis. Each mitotic division gives us 2 daughter cells, and the abdominal compartment, which we have already seen, the cell undergoes 2 mitotic divisions from 2 daughter cells to then form the secondary spermatocytes, and then initially and eventually to the 4 spermatids. This is the structure for it. We'll be discussing it in detail in later slides. So the transition of meiosis, when there is this transition of meiosis after the first mitotic proliferation division, this requires the cell migration towards the internal walls of the seminiferous tubules. So we have primary spermatocytes, which is a diploid set, which is 46. It contains 46 chromosomes, and then it enters meiosis 1. Then it immediately goes to the S phase, the synthetic phase, doubles their internal DNA and leave the basal compartment. As I said, it is the transition to meiosis, required cell migration. It leaves the basal compartment and then reach the special milieu of the luminal compartment. In meiosis, what happened? There is haploidization and then the genetic diversity to meiosis. Prophase 1 occurs, and prophase 1 is followed by different events. And these events further, what they do, there are different series of events which happen during the prophase 1 of the meiosis 1, which eventually does the reductional division for us. And there is pairing of homologous chromosomes. There is synapses via synaptonymal complexes, and then there is crossing over between the non-sister chromatids. So this is what happens, and then it finally reduces to the chromosome number 2 half. So then this transition is further taken to the meiosis 2, where we have secondary spermatocytes. Meiosis 2 give rise to spermatids, which are haploid and last only for 5 hours. Thus, the secondary spermatocytes are rather seen in the histological sections. So this is how it happens. These are the ploidy status of the different primary spermatocytes and the secondary spermatocytes. So the primary spermatocyte is a diploid cell. The secondary spermatocyte is a haploid cell. And then we have 4 spermatids, which are haploid in number. This is how we get our male gamete. Then we have the further last stage of the meiosis, in which we have 4 immature spermatids, and these spermatids, they contain nuclei which contain haploid set of chromosome. The autosome, it continues to direct the synthesis of the low level of RNA, mRNA, and the protein as they enter into the prolonged phase and the terminal differentiation, which is called spermiogenesis. So further, we are moving towards the second stage, which is spermiogenesis. Spermiogenesis is a cytodifferentiation event. Cytodifferentiation event. There are many things happening at a time in this, and what happens, there are many important events which take place. Like there is dramatic morphological modification, there is development of large site lysosomal structure, then there is nuclear remodulations and condensation, which is further, how does it happen? There are like series of events with 4 different phases. These 4 phases are Golgi phase, then we have cap phase, then acrosomal phase, and the maturation phase. So this is how it looks like when we have different phases for the maturation of oocytes or for the spermiogenesis, which is only the differentiation. There is no involvement of any sort of division in it. And first of all, we have the Golgi phase. The Golgi phase, what happens? The Golgi apparatus produce and release the lytic enzymes that are gathered inside the future acrosomal cap. Here you can see there is a cap formation over here. And in this picture also, there is like Golgi apparatus formed, and there we have proacrosomic granules which are formed, and they're finally changing their shape towards it. There is this gathering of the lytic enzyme, and also during this phase there is the genetic material packaging which is also happening side by side. This protamine replaces the histone. By the end of this phase, the genetic material is tightly packed and highly condensed. And then highly condensed, how does this happen? There is the nuclear condensation is required for the stability of the sperm DNA. So the histone is replaced by the transitional protein and then by the protamine. And then the supercoiled chromatin structure is formed to have a stable stable form of the DNA, which is highly condensed. So in the cross section, this is how it looks like. There was histone, which was finally replaced by the protamine in the further during the further process of the spermiogenesis. Then we have the cap phase. The cap phase is characterized by the events which leading to the formation of the acrosomal cap. In last picture, we have seen that there is an accumulation of the acrosome. There is an formation of acrosomal cap over here with which is very close to the nucleus, and a sac containing enzyme which is called hydrolytic enzyme are necessary for the oocyte penetration. It's starting to form very close to the nucleus here, and then further the elongation in these round cells, cells, they are they form an elongated structure over here, and it is formed when the Golgi apparatus surrounded the interiors of the spermatid to form the acrosome. This is how it is happening. It's elongating, then it is finally elongated, and then finally it takes out the head to the acrosomal cap. So then there is development of flagellum, and flagellum, there are in this picture, we can see that there are 2 centrioles. They are going to start to grow the microtubules, and that is going to be the base of the flagellum from here. But there are 2 centrioles, and these centrioles are forming the base of the flagellum over here. So the future axonemal structure, it grows out of one centriole, which is distal, and it consists of bundle of 9+2 pairing. And during this development, through rotation and nucleus, and acrosomal vessels and flagellum peridoneum contains to lie on the opposite side of the acrosome. So there are different parts of the sperm's neck. It have 2 centrioles. One is proximal and one is distal. Then we have midpiece. Then we have principal axis and the tail. Tail is having 9+2 structures of axonema. So then finally we have the maturation phase. In this phase, the residual cytoplasm has to be disposed of. Like in this picture, there is this excess cytoplasm which has to be disposed of by the process of phagocytosis, and by the process of phagocytosis. There is also this yellow organelle, which is mitochondria, which is started to aggregate and organize themselves around the proximal position of the flagellum, like this. like this, it started to aggregate here, and now it is formed over here in the mid-piece axis. This is the mitochondria which starts to aggregate, and this, after the maturation phase, this excess cytoplasm, residual cytoplasm, is being disposed of. The Serotoli cells and the epithelium cells of the seminiferous tubules that are having essential roles, that are also having the essential roles in the spermatogenesis. And during the early stages, they nourish the germ cells attached to them. So this is how the final structure of the sperm looks like after all these phases. It's like Golgi cap phase, then the acrosomal capping phase position, and then there is maturation where every cytoplasmic residue is removed. So finally, our sperm, it looks like this in the mature spermatozoa. This is the final structure of the sperm, and with all the labeling, there is like acrosome just above the nucleus, and then there is proximal centriole, there is mitochondria, we have axial phlegm, and then we have tail and terminal piece. There are other factors which can affect the spermatogenesis, which can affect the spermatogenesis very badly, and it can hamper the production of the sperm, which involves like there could be genetic factors, epigenetic factors, toxic components, and endocrinological imbalance could be there. So in vitro conditions, there are like, it is a 64 days of cycle, where the first meiosis, where the mitosis of the spermatogonium, it takes around 16 days up to the primary spermatocytes, and then the first meiosis, which is 24 days, second meiosis a few hours, and spermiogenesis takes around 24 days, and the total is 64 days. It takes around 64 to 70 days to form. Then this is how it looks under in vitro conditions. These are the Serotoli cells, and we have primary spermatocytes, secondary spermatocytes. This is how they look like under the microscope. Whenever there is like in vitro formation of it, there is round spermatids without tails, and then there is spermiation, and we have elongated spermatids formed in 14 days. So at the end, I just want to say that the spermatogenesis is a tightly regulated process that starts in the seminiferous tubule and ends in the functional terms in the female reproductive tract. So it does not it does not belong to the male only, and its functional terms, they get through the female, and then that only decides the functional capacity of a sperm, that if it is functionally fit or not. Thank you so much.
Thank you, Nancy. Do we have any questions?
Nancy, how does this protamination gives a stability to the sperm chromatin?
Sir, because the structure is highly condensed, and the histones are there, the protamines are more condensed than the histones. That's why it is replaced, but not all the protamine is replaced during this formation. It's like say 85% or 75% is replaced, and rest the histones are remaining over there.
Good.
So it is only for the audience. So audience can try whether they know the answer. Everything Nancy has covered very well in this lecture. So the first question is, primary spermatocyte undergoes dash to form the secondary spermatocyte. The options are mitosis, meiosis 1, meiosis 2, both mitosis and meiosis. So as I displayed the answer, so for the first question alone, the answer is B. So the correct option is meiosis 1. Coming to the second question, dash process by which the spermatids become mature spermatozoa. Dash process by which the spermatids become mature spermatozoa. Options are spermatogenesis, spermiogenesis, spermatid transformation, and the fourth option is maturation. The question is, dash is a process by which spermatids become mature spermatozoa. So the correct option is spermiogenesis. The answer B, option B. Coming to the third question. One spermatogonia give rise to dash spermatozoa. One spermatogonia give rise to dash spermatozoa. Let us take it in the way as primary spermatocyte, not spermatogonia. So one primary spermatocyte gives rise to dash mature spermatozoa. Options are 1, 2, 3, and 4. So the correct option will be answer option D, that is 4. So coming to the fourth question, phases of spermiogenesis are. I think this is one of the Eshrey questions in the year 2021. So phases of spermiogenesis are, you should remember the phases in the correct order. So the options are Golgi phase, cap phase, acrosomal phase, maturation phase. And the second option is cap phase, Golgi phase, acrosomal phase, and maturation phase. And the third option is acrosomal phase, cap phase, Golgi phase, and maturation phase. And the fourth option is acrosomal phase, cap phase, again acrosomal phase, and maturation phase. So the correct option is answer option A, that is Golgi phase, cap phase, then acrosomal phase, then last one is maturation phase. Coming to the last question for the first session, Leydig cells secrete dash. Leydig cells secrete dash. Options are estrogen. Option A is estrogen. B is inhibin. C is testosterone. D is FSH. Leydig cells secrete dash. Options are estrogen, inhibin, testosterone, and FSH. So coming to the answer, correct answer is option C, that is testosterone. So this completes the quiz session for the first talk. Let us continue with the second topic. Yoshita, you can start.
Ananta, thank you so much. Now for next session, I welcome Dr. Nandita Relekar. She is MSCE Clinical Embryology, University of Leeds, UK. She is a lab manager, ART Fertility Clinic, working in embryology since 2008. Her area of interest is male infertility and PGT research. Welcome, Nandita.
Yes, good evening, everybody, and thank you, Ahira, Rahul, and Retsa for inviting me for this lecture. The topic of my presentation today is grooming sperms for fertilization. After Nidhi has explained us how spermiogenesis is done and how the maturation process happens, let's now look at how to process the sperms for the best use of fertilization. Coming to the general discussion, the components of seminal plasma are essential for guiding sperms in vivo. In vitro, it's a different scenario. The same seminal vesicular fluid can have a negative impact on the sperm motility and DNA. Hence, it becomes essential to separate the sperm from the seminal plasma, and as suggested by WHO, within 1 hour of ejaculation for diagnostic, research, and clinical purposes. So what is the aim of processing this sperm? We need to process the sample to yield a final preparation of containing high percentage of morphologically normal and motile sperms with lower DNA damage. For clinical practice, the sperm should be free of debris, non-germ cells, and dead spermatozoa. Okay. Coming to the choice of method, the choice of sperm preparation technique is dictated by the nature of the semen sample and its purpose. An ideal sperm preparation technique should recover a highly functional sperm population that preserves the DNA and does not induce dysfunction through the production of ROS. It should be easy to perform and relatively inexpensive. The techniques for processing sperm ejaculates are based either centrifuge based or non-centrifuge based. We shall discuss each one in detail. For centrifuge based systems, before we discuss that, we discuss about a basic requirement that we need to have for all these processes. The semen sample should be collected with respect to the criteria mentioned in WHO. Preferably, we should have had the semen analysis done before the IVF cycle so as to avoid surprises on the day of pickup. We should have a backup sample for collection problems or if the partner is unavailable. Disposables should be non-embryotoxic and NEA tested. Reagents generally are balanced salt solutions containing protein and a buffer that supports environmental conditions in which the sperms are processed. Commercially available media is generally supplemented with HSA. In case it is not supplemented with HSA, the HSA needs to be supplemented with optimal concentration. The discussion to have CO2 or non-CO2 buffered media depends on the type of treatment, which is if it is only for ICSI or IUI, we can use a buffered media. But if it involves IVF as well, we need a CO2 based media and a CO2 environment that supports capacitation. Laboratory standardization, this is a very important part of stabilizing sperm preparation techniques. Reason being, number one, that we need to have SOP for sperm processing, mainly centrifuge speed and time, because this would change for each lab and each centrifuge machine as well. We should include the variation of samples. For instance, if we have a sample which is high viscosity, we need to discuss and decide, and up to the level of standard that we have available in our procedures, we need to know whether we increase the centrifugation time or we increase the centrifugation speed for a specific sample. The choice of method should be as per the type of sample, that is for normo, oligo, and actino, which we will discuss in detail now. Any modifications to these processes should be rigorously tested before the clinical implementation. Periodic testing should be done for determining the quality of the process, and functional capacity of the sperm must be checked periodically on the basis of motility and vitality. Coming to the first technique, which is centrifuge based, is simple washing. The sample types that can be processed by this technique is severe oligospermic sample, actinozoospermic samples, which can be used for ICSI. This involves only washing of the seminal plasma and resuspending the sperm samples for use. Generally, 1 or 2 washes are done depending on the sperm volume and the sperm count. Increasing the sperm media volume or increasing the centrifugation force or the time can be done depending on the volume and the count of each and every sample. The drawbacks for this method are they do not eliminate the non-viable sperms, leukocytes, or the debris in the sample. Coming to the next method, which is direct swim up. This is a sperm motility-based method, and it is the most elementary method to recover motile sperm based on their ability to swim out of the plasma and come into the culture medium. Most of the times, we use this for normozoospermic samples. This can be a direct swim up, which would involve raw semen and overlaid with a culture medium or a buffer. Or it could mean that you would first wash the sample with this media, remove the supernatant, and then add fresh media for the sample to swim up from this fraction of the pellet. Generally, it's kept at 45 degrees angle to increase the surface area for the sperms to swim up. The advantages are that it's easy to perform, it's cost effective. The disadvantages are that it has a low yield. The sperms can be damaged by ROS. Significant decrease in the percentage of normal sperms at the end of the process. The sample is not completely free of debris, and it is not for poor samples. Coming to the next and the most extensively used procedure, which is density gradient. It is most widely applied because of its efficacy and adaptability of the procedure. It is also easy to standardize in the lab. It provides a sample with a lesser yield, but highly motile sperm free from other cell types and debris. The samples that can be processed by density gradient are normozoospermia, teratozoospermia, and suboptimal sperm parameters as well. So the density gradient is a centrifugation of semen over density gradients consisting of colloidal silica covered with percol, which separates cells by density. It is generally followed by wash or a swim up. Commercially, density gradients are available as percentages that can be directly used, or we can use it from 100% and supplement it as per we require. The most widely used are either 80-40 or 45-80. The advantages are that it's a clean fraction free of debris. You get a good yield of higher on percentage of normal sperms and can be used for seropositive samples. The disadvantages are that the process time is longer, it is expensive, it is difficult for viscous samples or samples which are not broken down well by mechanical. It needs to be followed by other kinds of washing, and it has a less yield. So comparing the three methods, which are swim up, density gradient only, and density gradient and swim up, there are a lot of studies that say that when used in combination of both, that is density gradient and swim up, provides a better segregation of lesser DNA damaged sperms and enrichment of sperm with normal morphology and entanglement. Density gradient is also really better for samples which are teratospermic. There are so many studies that show that intact DNA or the DNA fragmentation after density gradient is much better in teratospermic samples. Coming to non-centrifuged techniques, first is magnetically activated cell sorting, which is based on the membrane characteristics. So for a general discussion, the outer membrane of the sperm is critical for its functionality as it is involved in many aspects of fertilization, such as capacitation, oocyte binding, and acrosome reaction. MAX takes advantage of these characteristics and allows selection of non-apoptotic sperm from a given sample. Now, phosphatidylserine is exposed on the outer membrane when the sperm is apoptotic. It involves the use of magnetic microspheres conjugated with annexin V that has an affinity for phosphatidylserine. When this is passed through a magnetic column, the apoptotic sperms are trapped inside the column and only the viable fraction is eluted. This is widely used for DNA fragmentation along with density gradient and samples that can be processed with MAX are normospermia or suboptimal sperm parameters. So this is a setup of MAX where you have the column from which the annexin mixed sperms are passed through. The ones which are trapped inside are the apoptotic sperms which are bound to the annexin, and the sperms which are eluted are the non-apoptotic sperm fraction which can be used for ICSI. So a couple of studies say that the spermatozoa prepared, a spermatozoa prepared showed a reduced level of apoptotic markers, suggest higher fertilization potential of processed spermatozoa. Integrating MAX as a part of sperm preparation technique will not improve sperm fertilizing potential to the extent of traditional sperm separation. So these were earlier studies that showed that there are half the studies that show that MAX is good. Some of them say MAX is not good. But MAX along with DG is proving to be much better. So the combination, so these studies are looking at DG followed by MAX or MAX followed by DG. So the combination of MAX followed by density gradient and swim up is of potential clinical value, which improves the sperm quality and semen preparation. By eliminating the apoptotic cells, the MAX DG technique optimizes sperm selection in the individuals with elevated DFI and better development of resulting embryos. Coming to another centrifuge-free technique is microfluidics, and this is becoming very popular nowadays. The reason being, as opposed to the traditional models and methods of processing, microfluidic chips and devices can be designed to create a bio-inspired environment for the sperm to increase selectivity, mimicking its in vivo behavior. It is cost-effective, it has a low processing time. Both methods are centrifuge-free, which is passive and active. The output is high and the reagent volumes are low. So today we are going to look at passive sperm selection because that does not include anything apart from just the device. The use of sperm motility, passive sperm selection uses sperm motility for separation. They do not need an external influence for sorting. Different models are available with parallel streams, spiral devices, membrane barriers. So there are a lot of sperm microfluidic devices available with a lot of complexity, but we will just look at two today. Both of these are extensively used. One which has a inlet where you add the raw sperm. There is another inlet for the media. And because of the laminar flow, the dead sperm is going to be guided to the end outlet. And there are bi-lanes for the motile sperm to separate themselves out from the poorer samples, from where it can be collected and it can be used for ICSI. The other one, and the more popular one, which is available now, is the membrane filter, where there is a sperm inlet. You add the media, and you have a collection chamber, which is on the other side of the membrane filter. So all the sperms that can pass through the membrane are the only ones which are collected. There are a lot of studies which have spoken about the DFI in these samples and have shown that the DFI for the samples produced by microfluidics are not different than the ones which are used in DG processing, but the progressive motility is very high. If you have a look at a couple of more studies which show that they calculated the DFI in the split samples for, and they looked at they have undetectable levels of DNA fragmentation in the ones which are microfluidic separated. There is another study that compared DG versus microfluidics in IVF cycles, and they concluded by saying microfluidic devices, which are labs on chips, are easy to use and an inexpensive method of sperm sorting. IVF success rate may improve with the use of microfluidic sperm sorting chip for sperm selection and male factor infertility. Processing sperm for seropositive samples, there has to be a pre-workup done for checking the viral load of the samples of the patients. And WHO suggests and talks about IVF samples where they use a combination of DG followed by swim up. And that is the proposed way of prevention of infection into the unaffected part. Prepared samples should be treated by RT-PCR before use, and only sperm free of HRV must be used. ARV therapy to reduce the viral load should be followed. Coming to surgical sperm retrieval and processing, sperm retrieved by surgical interventions due to clinical reasons may appear immotile due to lack of complete maturation. Hence, it becomes completely crucial to identify and select only viable spermatozoa for ICSI. For FNA, sperm concentrations need to be checked, and if there is a good amount of sperm available, either density gradient or single density can be used for processing these samples. Whereas where preparing TCA samples, there are two processes that can be used. One is mechanical process by glass slide, by macerating the sample and using the coagulum that you have found and then processing it. And the other one is by needles, by mashing the testicular tissue and then collecting this, followed by an enzymatic process, which is done by collagens. Once the sample is processed, you would make a slide and you would look at the samples. If you get twitching sperm, then that can be used for ICSI. In cases where there is immotility, we can have sperm processes done for checking the viability of the sperm. This can be done by various processes. The first one, which looks at sperm physiology for viability, is hypoosmotic swelling test. Tails of viable sperms swell and bend if introduced to a hypoosmotic solution. This is due to osmosensitivity of the calcium membrane channels. Several studies have shown that HOS can be used to estimate percentage of integrity in the chromatin. Several distinct patterns of swelling are related to the levels of chromatin integrity, allowing microscopic recognition for better selection of nuclear material. So here you mix the hypoosmotic solution with the prepared sperm solution, prepared sperm suspension, and then you look for the swelling. So if you have a look at the diagram, you have the sperm with no looping and the other one where you have the looping, and those are the ones which are viable sperms and that can be used for ICSI. Another way of checking the viability is introducing chemical inducers of motility. These are phosphodiesterase inhibitors, which are pentoxyphilin and theophylline, popularly used in IVF. They allow motility reactivation of spermatozoa recovered from testicular epididymal, as the maturation is incomplete before the epididymal, before the final tracts of the epididymis. So studies have shown that induced motility does not affect DNA damage or viability loss. So this is one study done by Vijay Manguliso, where they compared pentoxifillin with horse, and they proved that they concluded by saying pentoxifillin gives better clinical rates as compared to horse. There are also studies that show theophylline being much more superior than pentoxifillin, as pentoxifillin resulted in higher number of miscarriages. But in this study, the number of cases were very low. They were just 24 in. Coming to another way of checking the viability, lesser used, but this is one more way, and it's important for a certain reason. Laser irradiation causes the release of cytosol of secondary messengers such as calcium and increase in the synthesis of ATP, which will generate a slight movement of the tail. The tail coiling after a laser shot is considered as a sign of viability. So you give a very small pulse of a laser shot near the or on the tail, and you look for looping. So if you have a look at the diagram on the right side, the first one is where the shot is not given, and the second and the third is where the shot is given. Those are the sperms that you would use for ICSI. Now, this method is the last resort if horse or pentoxifillin has failed, only then. And in one very important reason, which is cartilaginous syndrome. In this, you have complete immortality of the sample due to gene mutations in the gene, because of gene mutations. And these are non-responsive to pentoxifillin or theophylline or horse. And that is when you would use laser to check. There has been this group that has reported live births of healthy triplets, 1.2 girls, by a double blastocyst transfer. Coming to retrograde ejaculation, as we know, the semen passes into the bladder at ejaculation, resulting in euspermia, no apparent ejaculate. Confirmation can be done by examining urine for sperms after ejaculation. The alkalization of urine by ingestion of sodium bicarbonate, for example, will increase the chance of any spermatozoa passing into the urine and retain their motility characteristics. Analysis of both ejaculate and urine samples need to be done. For larger volumes, centrifugation and concentration of sperm followed by appropriate treatment may be followed. In summary, each method has shown its advantages and disadvantages. The choice of method should be selected for each type to result in the optimal yield for clinical use. Our methodology should meet the quality standards of art to provide the highest quality of treatment. Newer research will open avenues for further discussion on the possibility of achieving a golden standard for sperm processing. Thank you.
Thank you, doctor. We have one question.
Yes.
Can you shed some light on the process of capacitation?
Very good evening, everyone. So moving toward the next session of the evening, which is a panel discussion on different case scenarios. We have with us our moderators for the evening, Dr. Rahul Sen. He's Chief Consultant Embryologist at Neelkant IVF, Rajasthan. He's Executive Member of ACE 2022 to 2024. He's also nominated as a mentor in ASHRAE Mentorship Program 22 to 23. And he is also the recipient of ISAR Shining Awards at Embryology ISAR 2023. So I welcome you, sir, on board.
Nancy.
Can I have the next CV? Then we have with us our next moderator, Mr. Samsher Ali. He is working since 13 years and currently designated as HOD at Department of IVF in Lifeline Infertility and Super Speciality Hospital, Kerala. He's also holding a position as Associate Faculty in Department of Biotechnology at National College, and awarded first prize for presentation at ACE Hyderabad. I welcome you, sir, and I request you all to please. I request you to please call our panelist for the evening.
Yes. Am I audible?
Yes, sir.
Okay. Thank you, Nancy, for this wonderful introduction. Taking the session forward, we are expecting Dr. Akraf Zakaria to join in soon. Yoshita, next CV, please. Dr.
Yes.
Mahiraj Singh Gaur, he's hailing from Rajasthan. He's also expected to join soon. He's an MBBS MC embryologist from Monash University, Australia, a very dear friend. Yoshita, next slide, please.
Uh, Dr. Nishad Chimote is also here, a dear friend of mine, and Nishad is having a vast experience of embryology and been a mentor for many of the budding embryologists practicing these days. And Nishad currently holds vice chairman place in ASAR. So welcome, Nishad. Next slide. Yes, Mr. Dilip Kumar from Bangalore. He's also here, a very dear friend. And Dilip is working as an associate director, embryology operations, Milan Fertility. And his area of interest and research are sperm kinetics and effect on male gametes and development on embryos and quality control in ERT. Welcome, Dilip. Another is Dr. Shilpa Marwa. She's pursuing PhD. She's working as a chief embryologist at Grace Fertility Center, Gurgaon. Welcome, Shilpa. And Yoshita itself, Yoshita, a dear friend. She is currently working as a clinical embryologist at Oasis Fertility in Bangalore. Welcome, Yoshita. Dr. Ananta Lakshmi, she is our iHera's very renowned program coordinator. Welcome, Dr. Ananta. She is the consultant and senior clinical embryologist at Janani Trichy Fertility Center. Welcome, Dr. Ananta.
Thank you, sir.
And Ashwani Dahale, she's a senior embryologist at ART Fertility Clinics in Mumbai. She is also expected to join soon. So I think we can start with the panel discussion. And Dr. Preeti Bista from Nepal. She is also a dear friend and a senior clinical embryologist, founding director at Angel Fertility Clinic in the Nepal International Fertility and Laparoscopy Maya IVF Center, located at Kathmandu. Dr. Preeti, please welcome and turn on your camera and voice. Along with me, my co-moderator for today is Dr. Shamsher Ali. So we both invite you for the extensive panel discussion on case scenarios of sperm selection for fertilization in IVF. So we have already discussed and seen about how the sperm uh, we have already been discussing throughout this evening how the sperm gets produced in the testes, the reloading and the protamine and the histone modification is there. Then the sperm goes maturation in the seminal plasma, and after that it basically involves into the process of fertilization where it faces a long series of events post ejaculation till the recognizing the zona pellucida of an oocyte. In the natural reproduction, the pathway is a very long path. In assisted reproduction, there are few of the steps which are skipped in the phase of reproduction. So we'll be discussing all these facts in this panel discussion. So I call upon my co-moderator, Dr. Shamshir. Can you take on this first question?
Yes, yes. So, as we know, we had previous discussion, two wonderful discussion. They were discussing about how important it is and what type of sperm preparation should be the choice, what type of sperm motility, everything is important. So, I would like to start with the basic question for all my. This question is for all my panelists. So, what criteria do you use for for a patient who's coming to you for IVF or XC? Do you have any set of parameters or criteria, something like that? Or what do you prefer? This patient should go for IVF or I'll do XC? I would start
Can I
with,
start?
yes, yes, please. Sure.
Okay, so obviously a really nice question, I think, and Nandita did mention a lot of things method-wise. But coming to the practical part of whether to select a patient for IVF XC, I have certain different kinds of criteria. Obviously, you mentioned about the motility morphology count. Yes, no doubt that is. But rather than that, I do DFI for all my patients, first of all. So if the DFI, because see, every lab has standardization, but if you use your dye method, if it is more than 15, it is more than 20, I would rather wait, give nutraceuticals and antioxidants and reduce it to 15 or less than 20 and then do even ICSI. One. Second, if the patient's age is above 40, the male, I will do ICSI. Third is your concentration and count. If it is maybe mild oligospermia, then we can think, but then you have to collaborate and also think about the motility. If it's teratosis, if it is SO80 even mild, then you can think about ICSI. But main thing is, even in that case, if you're getting good volume of semen, then you can make a good concentration with good grade 3, grade 4 sperms, because you require hardly 100,000 sperm, 200,000 sperms. So if that fits your criteria and your DAFI is less than 15 or just round about that, or even less, because sperm preparation methods actually decrease. I do post-wash. Post-wash, you do swim up, you do DG, you do microfluidics, you do any method, it will come down to less than 10.
Okay.
So that is not the thing. So if you're doing less centrifugation, getting a better DFI, maybe less than just around 5 or 7, then you can also do IVF. But age criteria, I feel, is a one important character.
Another question from your same thing. So do you prefer DNA fragmentation for all the patients?
Yeah, I do it mandatorily for all patients.
All patients.
Yeah.
Okay. So I would like to go for our next panelist. Shilpa, you can join and you can say a few words about your criteria also.
Even I also prefer doing DNA fragmentation. Even DNA fragmentation for all.
Okay.
And definitely for if I want to go for IVF, the scenario 1 where we have mentioned first cycle young couple or female is a normal responder, and the sperm concentration, motility and morphology is okay. The basic I go for morphology. The morphology is looking okay and it's a young couple, definitely I can go for IVF. Otherwise ICSI probably.
So DNA fragmentation, do you prefer the SC? Which method you prefer in DNA fragmentation test?
SCD.
SCD. Okay, good. So I would like to go for Dr. Dilip Kumar. Would you like to add some more points?
Yeah, Shamsi, yeah, fine. See, recently I go through the 2023 papers. It's shown that even the lesser than 1% of the morphology, they done the conventional IVF, and they shown the better fertilization rate than the ICSI. See, as we all know that the ICSI is only for the severe male factor, right? So all as of right now, all stick to the like all cases we are doing the ICSI only, but it's very clear indicated that it is only for the male factor. Previous history of the patient, then we need to go the ICSI rather than doing the like we can choose for the IVF. So I don't think like every patient we can go for the ICSI. Yeah, there is certain criteria we need to stick, and we can do the conventional IVF also rather than the ICSI.
Okay. Yoshita Tanwar, you can start with the first scenario. So
yeah, if the patient is normosospermia and everything is normal, so we can go for IVF, but we have to see the history of male patient also. If he has DNA fragmentation is high, and we can do microfluidics for that also in IVF. So we will get less DNA fragmentation, and for second scenario we will most probably we'll do ICSI only.
Okay.
Yoshita, can I just challenge your statement here? In a nice way, don't worry. See, you mentioned about microfluidics, post-wash DFI. If you do swim up, you'll have the same decrease in DFI, literally the same.
Sir, sometimes
yeah, good.
Sometimes we find the normal sperms, but it will reduce actually.
No,
And
the DFI comes down. I'm also saying that it's not only microfluidics. Actually, you can get it with normal swim up or also direct clearing. That is the opposite of swim up technique. If you do that, you'll get, for example, if the DFI is maybe around 15, after doing it decreases to 4, 5, something like that in both methods.
Okay, sir.
Basically, you do prefer microfluidics for those patients, isn't it, Yoshita? You
Yes,
do
sir.
prefer microfluidics, especially.
Yes, we are doing microfluidics for those
So,
patients.
like to go for IVF, right?
Yes,
First,
sir.
the IVF. Okay.
Yes.
And Ashwini, are you there? Ashwini
Ashwini, I think she's not joined.
and Dr. Anantalakshmi,
Yes.
you can go ahead with the second
Yeah,
scenario.
okay.
So
So
the
as previous panelists said, I usually don't do DFI for all cases. And basically the SED method which we are doing, the test itself can induce some sort of DNA damage in the sperms. So the gold standard is tunnel assay. So hence I don't believe, as such, personally, I don't believe in DNA fragmentation. So for the second scenario, for the first cycle, young couple, female is a normoresponder. And the borderline semen parameter means here I will take only the motility as a major criteria whether to go for IVF or ICSI. As Dr. Dilip said, it was not based on physiology. So if the motility is okay in the pre-wash sample and the count is okay, which is adequate for the oocytes which we get, I will go for IVF.
Okay.
Rather than I will go for ICSI.
So Preeti Bista, same second scenario. Would you like to add some more points?
Well, for the second one, I
So,
think,
yeah,
well,
me too.
for the first one, I think I'd rather go with the split one because if I have to add it on my practice, my center does not allow completely conventional IVF on any criteria of the patients. That's how the protocol is. So I'll always go for the split one because I do not want to take I don't want to risk if the hardened zona or if there is a fertilization failure or something. I always want to take chance because the female is a normal responder. For the second, I can see because all the parameters are compromised, probably I'll go with ICSI in all of the eggs. Probably incorporate with some sort of pixie, edge binding, or I'll probably go with the microfluidics as well.
So next slide.
Yeah, I'll.
Rahul, would you like to take?
Yes. Yeah. Yeah, you can
Rahul, Rahul and Shamsher, Dr. Zakaria is also joined.
Okay.
Well, I need
So yeah, we can
start
post
with,
this.
Dr. Zakaria with this question, I think.
Yeah, we can post. Dr. Zakaria, are we audible?
Hello.
Hello. Yes, yes. Welcome to this panel. So we were discussing this case scenario where the male partner and the female partner both were the normal responder, and a few of the panelists had commented that they will opt IVF as an method of fertilization. So after the fertilization has been done, there goes a total fertilization failure post IVF. So what can be the circumstances that you might follow in your conditions, or what want to say as your experience?
Okay, so thanks for the invitation. And I think the total fertilization failure post IVF is an indication for an ICSI, a microinjection, and especially it's called rescue ICSI. So rescue ICSI comes to rescue what we missed before. In this case, fertilization. So TFF or total fertilization failure, we proceed generally to an ICSI, an R ICSI, which means rescue ICSI. So this is my point of view. I don't know if you have another suggestions.
Yeah. So you have mentioned about rescue ICSI. I would like other panelists also to throw some highlights on the process of rescue ICSI. What time do you suggest? When do you suggest? Dr. Preeti, can we have your suggestions?
Okay, so rescue ICSI is the timing is so important. It's generally after 36 hours after the failure, and we proceed to ICSI. In some cases, there is success after ICSI. But total fertilization failure, let me say, it's an indication of non-male factor indications of ICSI, because we all know that ICSI is a male factor in the indication in the procedure, but nowadays we are talking about ICSI for non-male factor, and among all these non-male factor indications, we have the total fertilization rate. So in some cases, we use ICSI for this indication, and it's generally after 36 hours after the fertilization in our clinic.
Okay. Any other comments? Yeah. Yeah, Nishad, you can jump.
Okay, so I did this today. What I'm mixing matching stuff now. I used to do FERT check after 3 hours post insemination. Okay, so that actually, it takes hardly 30-35 minutes for the sperm to enter post insemination, and by 3 to 4 hours you can actually see the second polar body coming out. So that determines, because that is the right time to do if rescue ICSI. If you do it after 12 hours and 16 hours, it's not going to yield any result. It fails. So nowadays I do, like, I don't do it. I send my juniors, they check it, they send me the pictures, and that's fine. So this is okay, but now I found a better method to do it. If the oocyte number is less, then I will do ICSI straightforward, maybe 6 or 7 oocytes. Then I will not think, because less number means there is some oocyte dysmorphism. So that helps me to determine that factor. If I have more, maybe plus 10 plus oocytes, then what I do is at least 4 to 5 I do ICSI, rest I do IVF. So there is no chance of fertilization failure.
So
So we
again,
can use oocyte activation also.
But not in the first case. The oocyte is first cycle you can't. You can only do it when it has already failed.
Yes.
Right. So that is why. And second is I'm doing DF.
Failure. Yes.
Right. And I'm doing DFI for my center patients. So that's fine. But then I go to other centers. They don't do it. So for them, I have to do this.
Okay.
Yes, Dr. Zakaria, you were mentioning.
Yeah, I have one suggestion, is that recently we proceed to ICSI directly. It's just because of the fear of total fertilization failure. Clinics are using more and more ICSI just because of the fear of the frustration of the fertilization failure. So I think it's not a good idea to proceed to ICSI just because of the fear and the image of the clinic. So clinics are keeping to grow and to grow their image and avoid all sorts of failures. So just because of that, they are proceeding to ICSI directly without looking for the indication, the causes, what we can correct before proceeding to ICSI, etc. So total fertilization failure is an important subject to discuss it. Thanks for.
Dr. Zakaria, I completely agree with you. That is how it should be. That if we are doing IVF, you should go ahead with that. But many times, the centers in India, they have freelancers. So there is the reason where they have to do ICSI instead of IVF. That's the reason. But otherwise, I think IVF, many people have started doing more IVF nowadays, which wasn't the scenario 2-3 years ago. So things have improved, they have changed, and it's nice to see that more and more people in India are doing IVF as compared to ICSI also.
Yes, but don't you think that ICSI is a more risky procedure than IVF?
Absolutely. Let's nature do its work. Why should there should be any manual intervention? So IVF is better than ICSI any day. I would agree with that.
But
Dr.
it's not
Preeti,
for all the
would you like
cases.
to highlight something
One
on
more question in
rescue
this
ICSI?
ICSI. One more question for Nishad
Yeah,
and
yeah.
So who should understand this thing, IVF or ICSI? Either embryologist or clinician.
Actually, both.
Indian clinics, yeah. So most of the Indian clinics, it depends on the clinician. So they usually talk to a patient.
Clinicians.
When it comes to a practical, practically, let's imagine we are senior most people. When you say clinician will agree. What about the people who are in intermediate, let's say 4 or 5 years of experience? How do they go against a doctor, or how do you manage that particular thing?
See, for that, what I have noticed is you need to strengthen your theory part, your knowledge part. Because, see, any clinician will get convinced if you are able to convince them. You have the parameters. If you see the file, you discuss what is the age, what are the other parameters, how is the sperm, how is the oocyte. You show them the picture that after denuding, this is the oocyte or this is the cumulus. So if you discuss the real parameters, I think the clinicians can agree. The only thing is, yes, that it's like 3-4 years of experience, so embryologists are not taken seriously. But if they have a good conversation, a good communication, I think that can be solved easily. It's easier said than done, but then it can be solved.
Basically, this thing is coming to your lab KPI also. That also should be informed to the doctor previously. What is your IVF fertilization rate and blastocyst rate, and what is your ICSI fert rate and blast rate? That also should be conveyed. So, doctor, it is easy to convince doctor.
Main problem is that everybody is worried about fertilization rates. Nobody talks about day 3 and blastocyst. Let the fertilization rate come down, but see how many of them have become blastocyst from less fertilized. I think that should be basically taught to the clinicians and made aware. Everybody's like 80%, 70%. Are don't worry, 60 hai, to bhi 60 mein se how many of them become good blastocysts should be the end. Not
I
how
think
many
these days, I think these days the blastocyst actually has become a commercial parameters for the IVF. This is what IVF centers, mostly because, do you do blastocyst culture in your IVF lab or not? I think that has become a commercial parameters to advertise your IVF clinic. And because of that, I think there's an added pressure on the clinical embryologists as well. And I would agree with Dr. Nishad as well when he said that, most of the clinics are not talking about the day 3 and the day 5. I'm coming back to the question, actually, the total fertilization failure post IVF. So most of the time, I think it would be much easier for us to keep this in mind because it only accounts for 5% on average of total fertilization failure post IVF in all of the clinics that are doing it. So if you keep this in mind and do your initial workup properly, just as the clinician does their workup properly, if we do our workup properly as well, we are actually mitigating most of the risk even before it starts. So for an example, we are checking the sperm properly, the DFI, which actually I agree with the DFI practicing routinely because these days the IVF has become so common that most of the patients that come to our clinic, they have either already done some kind of a workup or at least basic treatments. So, and there is a strong history already for most of the, majority of the patients. So when you're running DFI, or in most of the cases, what I do is I like to go ahead with the karyotypic analysis as well. If there is bare minimum semen parameters or compromised semen parameters, I think that gives you an insight into how the IVF cycle is going to be proceeded ahead. So I think you have to prepare yourself way before the rescue ICSI day. That's what I would like to say.
So I like to add one more case here. So one of the case, like we got, I think 17 oocyte. In that, actually only 2 are cleaved. Out of 17, 2 are cleaved. Believe it or not, all that 2 will become the blastocyst, and she conceived. And when I look into the case totally, there is actually a lot of the DFI rate, as Nisha told that the DFI rate is very high, but luckily that patient is conceived. Out of 17, only 2 cleaved. We got a blastocyst. See, generally we need a blessed blastocyst that's going to be implanted. So it's not like how many numbers we get. It's not that. We need one embryo that is potentially for implantation. That is like we need to work it on. As the DFI Preeti told that DFI, Ishra is clearly told that it is not a routine, the DFI. It is only for the selected patient. We need to look into the patient age and previous history, and only selected patient we need to do the DFI.
Rahul, would you like to take?
Yeah, Shamshid. Okay. So, after jumping, seeing all the discussion regarding IVF, ICSI, whether which technique to choose, which not to choose, which to select. So, jumping on to the case scenarios, there are two couples which having wives with report all normal, means the wives were normal responder, and on the day of ovum pickup, one patient had the hematospermia and another patient had leukocytospermia, and the couples are not willing for oocyte freezing. So what will be the choice of procedures you will follow after seeing these kind of semen parameters? Would like to ask Shilpa, your comments on this.
Yes, sure. So first of all, I think I would counsel the patient that they are studies saying hematospermia and leukocytospermia have low fertilization potential and they have an effect on sperm quality too. So since patients, I will definitely try to counsel them and tell them to freeze for oocytes and for the next cycle, and just talk to urologists for their underlying infections and all. But if the patient is still not willing for the oocyte freezing, I will definitely go ahead with density gradient sperm preparation method and followed by ICSI.
Okay.
Yes, I agree with Dr. Shilpa. I will also go for double density, and I will also counsel for these scenarios. It will may reduce the blastocyst formation and fertilization rate and implantation rate.
Dilip, would you like to do something
Yeah,
different?
fine. See,
Yeah.
when it is emetospermia and leucotospermia, generally I don't like suggest for the oocyte freezing. See, basically, when we take the case on day two of the stimulation, we look into the case. I generally prefer sample. The patient should give the sample on day two, right before the start. We need to see that. So when we got the emetospermia and the leukocytospermia, that is a white blood cell and RBCs, we need to plan according to the cycle. It is not like on the day of the procedure when we see the sample and freezing the oocyte, that's not a right. Like even we need to look into the financial also. And on day two, we will take, in case if we get that, yeah, I'll go for the process. I'm going to do the as a double density. And even I can go for the microfluid method and I'll go for the ICSI instead of doing the oocyte freeze.
Yes, backup sample is needed actually.
Exactly. See, on the day two, we need to check that. I'm always say that on the day of the seeing the sample, instead of that, we can see on the once the stimulation started, we need to check the sample how it is, right? So that we need to prepare for that case. It is not that on the day we see and we'll that's freezing, that's not a right. We also do some homework, how the clinicians do. We need to do the homework and we need to prepare how to take this case forward.
I think I also agree with this. I think we should not start, or we should tell clinicians not to start any stimulation without semen analysis or without all the reports.
Exactly.
Yes.
So we need to, and if that cases, we can say that patient can go for the antioxidant so that they reduce some the contamination chances like emetospermia. And definitely in case if you get that sample on the day of procedure, instead of freezing, we need to look into the economically and even the oocyte freezing. I think is numbers also important. If you get one or two oocyte, I think we can't take the risk, right? Even the numbers of oocyte is important. Based on that, we need to take the call whether we need to freeze or we need to inject.
Yes, exactly.
And
But
I think
definitely there are the see as we are seeing there's a lot of technology in the market say that sperm sorter there is so many are in there. We can go ahead and we can take the call and we can do the ICSI. We process it and we can go ahead. Even if you do the like a sperm surgical methodology, you can see a lot of RBCs are there and a lot of pulses is there. We are processing that sample and we are doing the ICSI, right? I think in the similar way we need to, we can go ahead instead of freezing the oocyte.
Yeah. So, do you think in this particular scenario, having a second ejaculate, a simultaneous second ejaculate might work in this situation?
Definitely. In case if it is the in this like RBCs and WBC, not most of the cases. I don't think there is some the diagnostics required. There's some clinical call. I don't think it will be improved, but with that sample only we'll go ahead.
I think patient should be comfortable for giving another sample in just.
No, otherwise, sorry, actually, otherwise it is because you already seen that there is hematospermia or leukocytospermia. I don't think it will improve in second ejaculation. You have to go with the same sample. And if you want to go, then and definitely I agree with Dilip because you have to see the economics also. We should go with this sample. We have the techniques. We can process that sample like double density and swim up. We can do that, and easily we can go for ICSI directly, in my
So
opinion.
even like if you do the surgical sperm surgical,
Yeah,
we get
you
all
are
these
right.
things.
Yes.
Yeah,
We are processing that and we are doing the ICSI.
Yes.
So basically, the take home from this particular case scenario is like better not to have shockers on the day of ovum pickup or on the day of ICSI. Better to analyze, better to check, and better to have a thorough report of semen analysis just before starting or just before getting the patient into the course of IVF and ICSI. These conditions can occur at any time, at any instance. It can be due to injury also. It can be due to recurrent infections or fever or anything. It can be there. So it's better to get in touch with the patient, get all the details, not only of the semen analysis report, but their recent means behavior also, how they have been seeing in this all through the days of stimulation and everything. So yeah, next scenario.
Rahul,
Yes, sir.
because now I'm leaving on, I may not be able to join again. So just a little comment regarding the hematospermia.
Yeah. Yes, sir.
It is essential to get the information from the couple whether they have collected it by masturbation or was it coitus interruptus. So sometimes you may get blood contamination if the couple has collected it the other way. So please rule out whether they have collected it through the coitus interruptus. It may not be a true hematospermia, and you can just process the sperm with the density gradient and get rid of the RBCs.
Right. Absolutely right.
Yes, very, very well said, sir. Yes, sir. So,
Rahul, Ashwini is also joined.
Ashwini is there. Yes. Yes. Shamshir, can you take the next scenario?
Okay. So there is another case scenario. The man is 13-year-old and 3 consecutive essay report were cryptospermia. Wife is 30 years normal responder. So we have a different sperm selection method. So what do you prefer in this particular case? Would you like to go for fresh? We have frozen backup also for the sample. Or do you have any other thoughts about this particular crypto sample?
Yeah.
I would like
Ashwini, you can go ahead. Yeah.
Yes. So I would first see how were the sperm at the time of sperm analysis. So generally, what we should do in crypto is to take the pellet and search the pellet entirely under high power microscope. So get the idea how many sperms are. Hello, am I audible?
Yes, you
Yeah.
are audible.
So how many sperms are present? For example, if you see 2 sperms, 10 sperms per 10 microliter, just to have an idea. If it's a rare sperm, we have to keep this in mind. So we have to might not get any sperm at the time of pickup. So we have to plan the accordingly. Then there is a backup. There are 2 ways of backup sample. Just a simple straw, like just for backup. And if it's a rare sperm scenario, we have to use a techniques like sperm VD, where we can vitrify the individual sperms.
Okay.
And also, depending on the how many follicles the wife develops, we can plan schedule on the day of pickup. But this everything has to be scheduled a couple of days prior, not because if if there is a SOS TISA required, we have to keep everything ready. So if there are so if you can see a sufficient number of sperms available as per our oocyte count, we can just will require 1 sample, and it will be okay with the sperm just dish and normal ICSI. But if you require more sperms, you might need a SOS TC also.
Okay. Yoshita, you're adding some points. You're saying something, Yoshita?
Dr. Ananta,
Yes, sir.
Anantalakshmi.
Yeah, I will also go with Dr. Ashwini. As she said, sperm vitrification is a suitable method for very few sperms when it is available. So if it is not adequate for the oocyte, we can try with TSA. Otherwise, I think I will go with a fresh sample when it is adequate.
Okay.
Yes, but it should have one or two backup for
okay, more back.
yeah.
So Nishad, what is your thoughts on this?
Yeah, obviously the same thing. Surgically, sperm retrieval will be a better. TISA method will be much better. And after TISA also, that whatever sperms are retrieved, I'll use sperm VD then. So I have backup for the next cycle in case it first one fails.
So let's say that critical spermia, all the sperms are abnormal, all are head, tail lift, all the defects you could see. There is no not even motility, twitching, nothing. So in that sample, what do you prefer?
Right. So in that, see, first of all, this was only the first part of it. Now what you're saying makes more sense. That in such a scenario, what I will do is one thing is host. So you have culture water. Okay, 1:1, do it, and the one which coils is the sperm that is vital, which is alive, has a DNA kind of intact, kind of, not exactly. Fine. So I'll inject that sperm because that is alive. So that will give fertilization, and further, if it's good quality DNA, it can become a good embryo or a blastocyst later on.
So do you prefer fresh or would you like to go for TISA sample or TC
See,
something?
considering the right now the techniques and the sperm survival and what the vitrification media can do to the DNA of the sperm, because generally oocyte is having a different kind of DNA genome structure. Here we are talking about protamine and 2 dumbbell shape, which is more fragile. So there is a higher chance of that DNA getting damaged because of vitrification. So I would rather go in for a fresh TISA sample rather than a vitrified sperm.
Fresh sample also you see only defected sperms. So do you prefer TISA
so
or
fresh sample, obviously, I would rather go for surgical retrieve sperm.
Okay. Anantalakshmi, in that case, what do you do in morphologically all abnormal you see on the day of, in normal essay or everywhere?
Not only based on morphology, I will decide for surgical sperm retrieval. I will go with the fresh sample.
So even though the sample is having very poor morphology?
Yeah, I prefer to do with the ejaculate sample.
Okay. So don't you think that TISA would help with these samples?
Sometimes, as our andrologist, we have faced the same scenario in our clinics also. He is saying that ejaculate sample are more better, and only for the DNA fragmentation, even if you take the studies comparing the TISA sample along with the fresh sample, there are more studies against this also. Fresh sample is better. So I prefer fresh sample rather than TISA.
Okay.
You may or you may not get samples in the TISA as he ejaculated, and the production itself is very less. So we may land up with immature cells with the TISA.
In case of TISA, let's say you have done the TISA for some reason, there is no motile sperm, no twitching, nothing. So do you prefer any solutions or is there any?
As he said, I prefer HOS, or I'm also used with the theophylline and pentoxyphylline to finding the twitching moment.
Yes, Shamshir, just to answer the question, I get your point here. When it comes to theophylline or pentoxyphylline, it's now well known that pentoxyphylline is more toxic. And I think Nandita also mentioned in her paper, Vijay sir has done his work regarding that yes, pentoxyphylline works very well as compared to HOS. But considering today's scenario, theophylline is the safest and the best method if you're not opting for HOS.
Okay. So the conclusion is mostly we'll go for fresh sample. And in case of no motility, first we have to go for HOS. In case of no HOS, then theophylline is safer, right?
Yeah.
You can use laser also to check when there is live or not live in case of non-motile sperms.
Interesting thing, how many of you are using laser practically today?
I will disagree. I have done it very early.
Say we should have
no, you do.
No, I'm just asking.
But actually, the piezo ICSI is right now trend that people using the piezo ICSI that gives the more fertilization. Ishal. So there is a lot of paper says that the piezo ICSI is giving the good fertilization.
PISA is you're saying which one? The laser.
Piezo ICSI.
No, the piezo. I'm talking laser hit.
Laser. No, but
laser does not work.
Dilip, do you think the piezo ICSI has its worth in it? Means it's a very new technique and not much research or not.
Yeah, what
publications
about
are there?
center who don't have a laser? The laser costs nearly 20-22 lakhs, so it's not affordable for everyone, right? So
I think
by
it's just an
the
excuse to use laser one more time. You should not use
Yeah,
laser for
by
sperm.
I
I'm
think
being very
HOS
clear
is
on
the
that.
best, I think.
okay,
no, I think even by the injecting pipette, you can make sure that whether there's any motif like twitching in the sperm. We
yeah,
can make
but
sure that we can touch the tile. We can see that. So that touch the tile and we can make sure any the movement over the tile and we can select the sperm.
first you go and touch the sperm
That
and
actually technique we can use that instead if there's no laser.
okay then.
Yeah, I think that would be the best. The mechanical touch
Yeah.
works like 9
It
out
does
of 10
work. It
times
does
it
work. Yeah.
works. Yes. And just the
Information.
increased incubation time as well. And if it's not working, just put it in the incubator under the oil for like an hour and a half and then see, you're gonna see like some of the movements and then you can just load it for the ICSI.
Exactly. Okay then.
Yeah.
Message for the junior Cs, just first go tap the sperm and just wait and see whether it is moving or making a twitch. Otherwise, do a horse and then go for theophylline step by step.
Yes,
So
yes.
next slide,
I think Dr. Zakaria want to say something.
Zakaria.
Yeah, so I just have one comment. I don't know your practices in India, so I have no idea about it, but I will just talk about my experience in our country. So can you please come back to the case?
Backslide.
Yeah.
Yeah. So 3 consecutive semen analysis reports. So in our practice, we are working in an andrology lab as well. So 3 consecutive semen analysis already from the second semen analysis, the laboratory is recommending and it will be mentioning that that the clinician should think about sperm freezing. So just we see when we see that there is a case of cryptospermia, which means after centrifugation we get a pellet with 2 or 3 in our practice we have 2 or 3 2-6 max spermatozoa. So we mentioned in the semen analysis report that in this patient we should think about sperm freezing, because if it's not the case, we will have azoospermia after that.
So that's what. So we also prefer sperm freezing, not only for crypto, for all. I think all of us are following sperm backup for IVF all cases, if I'm not wrong. Everybody.
Yeah, and I'm okay. I agree with the hostess. And in this case scenario, what we are using is surgical sperm retrieval.
So one more question to ask, especially in this case of cryptospermia, what is the survival rate of the sperm? Do you go for a traditional method of sperm freezing, or do you use any special method for this kind of sperm freezing there?
No, it's just a traditional one. Yes,
So
traditional sperm freezing using micro-rotation.
again, it's a cryptospermia. Is there any particular volume do you freeze?
Pardon?
It's a crypto sample. So do you prefer any volume to freeze, like less volume, more volume? How do you freeze those multiple samples?
We have devices of single spermatozoa freezing, and this is what we do usually in our practice, but using micro-rotation.
Okay. So next slide, please. Rahul, would you like to?
Yes, yeah, you can take it.
So there's another case scenario. So patient is 34-year of man with primary testicular failure, resulting in maturation arrest, not willing for sperm donation. Wife is good responder, not willing for oocyte freezing. So how do you proceed? So this I would like to ask Zakaria again to Dr. Zakaria.
Yeah, I will think about surgical technique.
Okay. So Preeti Bista, would you like to add some points for this case?
Um,
Sample.
I think this calls for the urology consultancy. I think I'll first try and find out whether the reason for the primary testicular failure is permanent or temporary. If the patient is taking any kind of medications, probably do a karyotyping. And then what my urologist routinely does is a SCOS test for a zoospermic patient or in such cases. If we see that, we can alter the results by maybe administrating TTRT in these kind of patients, like testicular testosterone replacement therapy of some kind. But again, I'm not sure if this is during the stimulation the question is being asked, because then there would be like very short time to do anything. But if history with such patient will come to me, we will not start the stimulation until we have treated the husband first. And maybe after every 3 months, we'll just go a repeat testing for the Sertoli cells, and maybe, we can just keep on adding the testosterone until we see something, and then it would be followed by the testicular extraction, and then we go for the ICSI. Yeah, so I think that's what I would like to do.
Mahira,
Regarding,
what?
can I answer?
Yeah, you can.
Yes, please.
Regarding maturation arrest, you can classify the case as early maturation arrest and late maturation arrest. So if the patient is having early maturation based on after doing the surgical sperm retrieval, then only you can classify. So based on the cells available during the surgical retrieval, we can make use of that immature cells to do the ICSI. Whether we get round spermatids or the spermatocytes, we can use that for ICSI. If we fail with the routine testicular extraction, we can try with micro TCA also.
Okay.
And I think these kind of patients, even though they're not willing for sperm donation, I think what we always ask them is to, be ready for sperm donation. We do counseling sessions as well, because sometimes even after the repeated FNAC, and then you go for SCOS, and whether or not the testicular extraction would result in getting good embryos, I think we always give them this option that, they can either go for the sperm donor.
Okay, Dr. Zakaria, you would like to add some points?
Yes, just about the case that you mentioned about people who are taking testosterone and steroids hormones to get the muscles, etc. And in this case, we will get maturation arrest, but it's temporary. So we proceed to an hormone treatment using letrozole or anastrozole, just to get back our spermatogenesis, and minimum of 1 year or minimum 6 months to have our next semen analysis. And we hope for getting a spermatozoa, because it was just about taking all these kind of steroids, etc. So you should go to medical therapy first, and after that, if it fails, we proceed to testicular sperm extraction.
Same scenario, what embryologists should be doing in this kind of cases? What they have to improve, or do we improve by adding something?
You're asking me?
Yeah, Nishad, you.
Okay, it's the same question, the extension you're saying, right? Actually, I was just about to say what Dr. Zakaria said, that mainly this problem is more of a clinical than a lab problem.
Yeah.
First of all, your HRT, your physical examination, follow up with SA, whether it's increasing or not, this has to be done for 6 months. And only after that, you can go in for that. And the best option for such, because suddenly miracle is not going to happen and you're not going to get a normospermic sample. So the best option is, first of all, you do the basic analysis, semen analysis, and obviously if patient, and see, here it's not only the physiological problem, here the psychological, the male ego is also there. So here the patient wouldn't mind doing, we are talking about India here. So the patient wouldn't mind going for DFI also. So with this, why I'm saying this, because this helps for the counseling of the patient also. So these parameters will help you to determine whether you should go in for ICSI or not, whether after the HRT, testosterone therapy, and the rest, letrozole and everything, you can either go in for TISA, but that will only be after regular semen analysis reports. So patient to patient, it will differ. So some patient you might need to require to do TISA and do ICSI. Some patient you'll have to do with the sample you're getting sperms. You can directly go in for ICSI.
Okay.
Can I say something here? I have a question for Nishad.
Yeah.
Yeah, you just said that, giving HRT and will solve this problem, but this is a primary testicular failure and not the case of we have to like it's definitely a clinician's job, like clinician's part
Yeah.
in this case, but we have to check the FSH level also before starting the HRT, because if it's
Yeah.
high, the
So that
HRT
is why
is
I'm saying
not
it's a
going to work.
clinical part totally.
Yeah, no. I'm just telling if it's a primary failure, so we have
Yeah.
to only have one option of micro TSA,
See,
because the HRT will not work in this case if the
see
FSH
first,
is
yeah.
really low.
See, it's not only FSH now.
Yeah.
We have to also check for inhibin B. That is a major factor here for such patients. First of all. So that will be another factor, because hormone endocrinology plays an important. Generally, a primary failure is related to hypogonadism. So first is the treatment of the
Yeah, that is
hypogonadism.
why.
Yeah. So
Yeah,
you
if it's
treat
a
hypogonadism.
hypo,
Yeah.
yeah, if it's a hypo, then we can treat with HRT. But I think, just correct me if I'm wrong, if it's a complete failure, if the FSH is already high, then we cannot do anything in with HRT, or we can.
See,
I
that will
have a case in which the urologist had actually sent the investigation for the mutation, the AGF mutation. And based on that as well, they decided whether or not they want to proceed ahead with the TTRT, the HRT for the man. I'm not sure if that's in practice in India, but I'd seen that in one of my patients. So that, I think, is also a determinant factor that whether the treatment is going to work or not.
So we are
That helps.
More on clinical set. Shall we go for the next slide, please?
Yes.
So the next case.
Yeah. So there is again next case scenario where a 37-year-old man with having severe oligosteatoteratosis samples, and there has already been 2 previous ICSI cycle failure with low fertilization rate and poor embryonic outcomes. So and again the wife parameters are the normal onto the normal side. So, do you still recommend the DNA fragmentation in this particular scenario? What will be your process of fertilization, whether to go with the traditional ICSI or any other methods of advanced ICSI versions like IMSI, PICSI can be incorporated in this situation? I would like opinions from Ashwani and Dr. Ananta.
From my side, it is a severe oath case. So after 2 cycles of ICSI failures, even after that, I don't go for DFI. But in order to counsel the patient, if they ask why, what is the reason for this, in order to counsel them as such, we can do DFI, and we can contribute the probable reason may be that. And coming to the strategy for ICSI, any advanced method of sperm selection, we can actually, it is a severe oat, we can go directly with the direct swim up is a simple method which is very helpful. If you want any advanced method, we can just go with the microfluidics.
Okay. Yeah, Ashwani, yes.
Yeah, so I would like to add, we can also take the second sample on the day of pickup to reduce the DFI. And we have to check if the motility is
No,
less, if the sample is...
there is not any DFI done in this particular scenario. It's basically just to ask you that if or in this particular situation, whether a DFI can be a problem in this situation, or whether to rule it out or not to rule it out. Yeah.
I agree with doctor what she said earlier. So I will go with the advanced techniques here. Like if the motility is not the issue,
but it is here. So I will go with the sperm mobile if the severe steno, and also oocyte activation. If it's a total, non-motile sperms, then I will also go with the oocyte activation, like calcium ionophore.
Yes, Nishad, yes, would like
Two
your
previously failed
opinions.
ICSI cycles, right? So
Yes.
it still has low fertilization rate. DFI, you can do it, but the point is, again, I'll say that first of all, uh, suggest tell the male to go to the andrologist first, because this seems to be more of a problem to be sorted on a physical part, whether the SOAT is related and can be cured or at least taken care of, first of all. If that is the scenario and if it can be taken care, then we can definitely go in for the ICSI strategies and sperm selection methods. So that will be my approach in the beginning, that since 2 cycles have failed and if in their history they have not consulted an andrologist, I would do first that. Second is DFI, yes, because it will help, because since it is not done, at least I can rule out that the fragmentation index can be a factor. If it is not a factor, then it might be a physical factor. So that is easier for me to tell the patient to counsel patient. So we can rule out one factor by one factors, we can rule them out.
So do you think getting a karyotype done in this situation or in this scenario can be a help
Why
to rule out?
microdeletion can be done in this patient?
Yes.
Yeah.
And any strategic method for fertilization as of recommendation?
Well, rather, the thing is, with normal methods also you can get sperms. SOAT, if you're getting grade 3, grade 4, decent looking morphological sperm, that is the maximum an embryologist can do. So any method, whether it's a microfluidics, whether it is swim up, whether it's density gradient, yes, you can opt for your method of choice. No doubt. Because there are no papers in the world which say that this method is better than that method. So it is to each
Nishad,
their own, basically.
I'll add for this. Okay.
Yes,
Yeah.
So
yes,
as you
Nandita.
said that, yeah, definitely we need to do the DFI. I think if you go for advanced method, somewhere we can reduce the like a DFI rate, or what do you say, the abnormality is in the genetical level. So that definitely it is in embryology lab, which sperm you're going to select is the matter. So even though if you do the DFI, whatever it is, finally the embryology, we're going to select the sperm and inject, that is going to the matter. Probably by doing the advanced method, we're going to reduce the DFI level or abnormality, and probably we're going to select the better sperm rather than we are doing the like traditionally we're going to select. That's the one. I think nowadays we are seeing that centrifugational force going to give the most stress to the spermatozoa, so that we're going to use the microfluid. So we need to avoid the more centrifugation force. Yeah.
Right. So now the question is how much of centrifugation force? The problem is we are following WHO. It says 18 minutes and 16 minutes and 15 minutes, 1500 RPM. First of all, if you're making your sample with basic simple swim up method at less than 300 Gs for 8 minutes, you can get good swim up, good sperms. So there is no effect of centrifugation on the DFI in the first place. And second thing is, this is confirmed method. Do post wash DFI. If your DFI is less by normal method, you don't need an advanced method. You can just use those pumps. So you can confirm that my DFI is less. Maybe it's 5, 6, 3, 4, whatever it is. Now I can inject this sperm even with the simplest of the methods. So whether you do swim up, you do max, you do this microfluidics, they yield the same result. That's what I'm saying. It doesn't
So,
advanced
like
method, they
instead
give
I'm
the
saying
same
like
result.
instead of
DFI is always less in all of them.
instead of doing the
It's a marketing
centrifuge,
gimmick. I'll be very clear.
no, it's not
Yeah.
instead of
I just
centrifuge.
want to
No, I'm
add
saying
one
that
point,
if
Nishad.
sample is good, we can do the underlay instead of the centrifugation. We can take the supernatant, so then we can give the like limited centrifugation, so that will give the good fertilization. What I am doing. So even I'm not using the advanced methodology, but the some cases I'm doing the underlay. I'm not doing the centrifuge, right? So in that I can take the supernatant, and in case it's required, I do centrifuge, or I can select the sperm.
Yeah. See, because
Yes.
we are
And
required only about
also,
10 handful of sperms. So you can get it
yeah,
just like that. We just
Exactly.
have to
and
mix it. Yeah, Harsh.
I agree. Even I would like to add here, the microfluidics will not work if it's a severe OAT, because for
Yes,
example, there is a membrane, and if the sperms are good motility, then only they can swim up. If it is a severe OTA, the microfluidics will not work in this case.
Yes, Shilpa, you were mentioning something in this.
Yeah. First thing, I just want to add that though it's a clinical point of view, or it's a clinical part, we can also put a patient on medications for fertility enhancers. There are certain fertility enhancers available in the market. We can put patient on that, and definitely it is increasing motility and the quality of sperm. I have personally saw that. And second, I think if it is severe OATS, probably after getting the sample, we should do ICSI as soon as possible. Otherwise, we'll lose those sperms also if we go for DNA fragmentation post wash. Probably this is my point of view.
Actually, Shilpa, as you told that you have to start fertility treatment. In my view, without doing DFI and without doing ROS, I think don't put patient on the treatment or any antioxidant.
No, this is after
Because
DFI.
this is not good. Because now studies are coming that by giving antioxidant empirically, they reduce the telomere length. So we have to very sure about that before starting any kind of treatment with antioxidant, that is there any problem with ROS or DFI. If it is present, then definitely you can start. Otherwise, don't do. And don't prolong the treatment, like not more than 3 months. You have to check it again. Then go for further. Otherwise, don't do the treatment like that. It is my personal opinion because I am doing infertility also, male infertility also. So I have checked it. And recently in Delhi, Dr. What is the name of that lady? Rima Dada. She was there and she presented in her oration that by giving antioxidant, it reduces the length of telomeres. So we have to take care about that also before starting any antioxidant.
Okay.
Yes.
So I have one more point to ask for all the panelists. Anyone can answer, especially in this cases, going for advanced technique like max microfluidic. Do you think the daily ejaculation will help slightly to reduce the DFI, and then we can go for fertility and do ICSI? So
Yes,
daily or alternate, I think would work.
My question for all the panelists, anyone can answer. So when it comes to DNA fragmentation, why are we directly jumping to microfluidics or max? Why can't we just prefer, if the count is normal or slightly SYAT, why can't we go for daily ejaculation and we get slightly better sperms and we can go for
we are doing that also.
Right?
So
I already
how I do it is, I'm sorry.
Yes.
Yeah, you can.
So how I do it is as soon as I have my reports in the previous cycle, whenever the couple walks in, that's when we do all the tests. And I kind of put them on a timetable. I just mark their calendar saying these are the dates. And even if you miss a couple of them, because that would be too much pressure to them. So I just say that these are the dates and this is where, the ejaculation should happen. And then what I do is I just put them on 48 hours of abstinence right before the ICSI day. So anywhere from 24 hour to 48 if the DFI is more than 25. So this is what I've been doing. And before that, they can do a single day alternate or a double day alternate. And this goes on as soon as the ovarian stimulation stops for their patients. So this has actually helped a lot, whenever I do the resulting DFI, sometimes just to test whether or not it worked. And it actually helped in decreasing the DFI from 10 to 15%. So that also works. But then I also try and do PICSI or microfluidics routinely for most of my patients who ever come after the RPL and RIF. In this question, I'm not sure why we're not talking about karyotyping. And we are more focusing on DFI because if the patient already has 2 failed ICSIs, I would personally not proceed without genetic intervention because there could be a condition like 9QH plus or pericentric inversion, which can lead to these kind of results. So I think not just a urologist consultancy, but also a genetic counseling and a genetic intervention is required, even before you just ask the patient to come on day 2 and restart your stimulation. I think you need to take some time off between the cycles and then start to do the workup and then only proceed further.
Dr. Zakaria?
Yeah, Dr. Zakaria. Yeah.
So I just have one comment about the DFI or the SDF. If it's not done, is it really recommended? Because I have here severe OAT, oligosteroteratosospermia, and in andrology lab we recommend a minimum of 1 million of spermatozoa per milliliter to assess the DNA of the sperm. So if we have less than 1 million, we can't even run the test of the DFI. So how can we proceed to these tests? I agree that we should assess the sperm DNA fragmentation because of the age of the man, which is 37 years old. Yes, there is a probability to have a high DNA fragmentation, but I don't know if the percentage of the fragmentation, I think it will be high because it's a percentage, and we will have in the assessment we will have not enough spermatozoa to assess and to give a correct percentage of the DFI. I don't know what's your opinion about.
Okay. Any other panelists would like to add some more points?
So I would like to add here. So in such scenarios, what we ask patient is to give 2 ejaculates, and we use the latest one for the ICSI.
Okay, on the day of ICSI.
Yes.
What time difference do you think there should be between 2 ejaculates?
So what we do just at the time of pickup or before the pickup, we give the containers for the first sample, and at the time of denudation, or because we do denudate, we have this fixed SOP. So maybe after 2 hours before ICSI, we give the second container. Or if the patient is unable, patient has a history of difficult ejaculation, we already we counsel them to do a previous night ejaculate and come. So just to reduce the DFIs.
Okay, so just to ask the other panelists also whether to have ejaculate two double ejaculates on the same day. Do you think it can be recommended as ejaculations on every day, like just after the giving trigger, means the when the trigger is mentioned to the female partner, the male partner can also be recommended that it should be done as ejaculate on every day, means on the day of trigger, the next day, on the day of ovum pickup. Do you think multiple ejaculates on every day versus the double ejaculate on the day of ovum pickup, does it make any difference?
Rahul, I don't think any
You also
makes any
prefer
difference
double
here.
ejaculate.
Yeah.
I also prefer double ejaculate rather than it may be very difficult to have multiple ejaculates. So as Ashwini said, I prefer with double ejaculate, and we also advise the same. On the day of pickup, we ask them to ejaculate there and come and give the sample, fresh sample here.
Okay.
But sometimes patient not able to give sample second time.
That is the major
same
concern, that
day, sometime
on the
it
same
become
day
very,
getting
Yes.
that double
It is
ejaculate.
difficult. Hmm.
become
So
difficult.
that's actually more stress to the patient. Like if you ask a double ejaculate, I don't know, like some patient can give that, some patient maybe not.
Yes.
So with that, we can, instead of repeated, we'll go for the multiple samples. That will be good.
Yes.
Like what Preeti said,
Usually-
we also do the same thing. That I mean, you can go in for the double ejaculate, but on the same day. But it is safer that you tell the patient to ejaculate 2 days before, 48 hours, so that at least if there is infection because he's not ejaculated more than 7-8 days, better to get it done then, and then next time you can take 2 ejaculates.
And before OPU, we can take backup sample with alternative ejaculations.
Before OPU, what?
Backup sample. She's talking about backup sample.
Backup samples.
Sample should be.
like a frozen sample.
With alternative ejaculations.
Okay. Shall we go for next?
Yeah, next. Yes, you can take it.
Shamshir, can you go in previous slide?
Yes.
As you mentioned in second point, two previous failed ICSI cycle because of low fertilization and poor quality embryo at other clinic. But nobody discuss about the oocyte activation. There already low fertilization is happened. Can we use the oocyte activation in this? Fertilization is there.
So how many of you would recommend oocyte activation in this case?
See, if some of them are fertilized, then oocyte activation is not a factor.
No, because only low fertilization is there. No? Not all
Sir,
fertilized.
Exactly. I agree. But the thing is that
if there are 15, 16 oocytes are there, all are matured, and we got only two fertilized oocyte, maybe
right.
gamete. So
right.
what you will recommend?
So that is why the DFI is not done is going to help.
But DFI in this case,
we do the
DFI is not
DFI.
possible.
Right.
But in this case,
oocyte activation is not. So oocyte activation is not required when it is a low fertilization and poor quality, that too severe oats. I think oocyte activation is not required. Here we need to purely look into the male factor.
The oocyte activation, two things I would like to say is, first of all, it is still amber colored. It is still experimental one. Second is generally oocyte activation, like what we practice is only done when it is total fertilization failure. So I would not like to take in some experimental treatment which does not give you 100% result just for the sake of the third cycle. I would rather go in for a DFI and then determine and then think of it.
Because recently I saw one paper in, I don't know, I forgot that paper. They have done the study on that low in poor fertilization also, and they got the good result out of that. I forgot. I will share with you people.
Sure. I would love to know that, sir.
Yeah. It is from European group. I will share with you.
Yeah.
Next slide, please. So another case scenario, 32 years man with normal karyotype, 26 years woman with normal karyotype, married life 3 years, primary infertility, severe asthenospermia, and the complete absence of motile sperm on the day of ICSI. So what do you prefer in this kind of cases?
Normal.
I think Nishad has already given answer to this.
I think we probably discussed all of these questions in the previous question, right?
We already answered this. Pentoxifilin is toxic. Hence, if we after horse, we have to go for.
Okay, then I'll change the question. All of you. So there is second question, sperm preparation method. So in case of TISA or micro TISA, what do you prefer? How do you prepare your sample? So this would be helpful for the juniors who are watching this. One by one. Nishad, would you like to start?
Yeah, I think rather than me, I think Nandita's talk was very good regarding how you have to deal with this. So micro TISA, there are two methods. One is when you macerate the sample, right? So you can just tease it with the needle, right? So and separate the seminiferous tubules. You can open it and then macerate it and then centrifuge it. Obviously by basic density gradient method. Also single density here because double will not help. One, in addition to that, you can add collagenase.
Okay.
So that will break down the rest of the particles and cells and RBCs also in some cases, so that the rest of the only you have to get the sperm in such a case. So that helps. So combination of both will help you to get a better sample because TC sample, TISA sample are slightly tricky to obtain. And first of all, when RBCs and the debris is there, you are not able to see. Adding collagenase to that is actually saves you almost like 2-3 hours of work.
Okay, so you do prefer single density before for preparing the TISA sample?
Yeah, mini per call. I mean, that was what we used to call early times. Now we just call it single density. So that, but now in such cases, yes, if you're getting, maybe you can go in for other methods if people are using that. I'm nothing against any method.
So any other panelists who are using different than this particular method, they can add the points.
Yeah, we are using collagenase and ELB. but the protocols requires a lot of time. So it is not one can plan at the start of the pickup day, let's do this. It is not possible. So it has to be a schedule. It has to be well planned.
Okay. So any other preparation method? Anybody using different method?
No, I think I do exactly what Nishad does. It's just that I try and schedule the procedure like 3 to 4 hours before the pickup, and then I would rather then just incubate the sperms that I found in the sample, and then I just keep them just to make sure if they're not moving or they're not motile. So during the ICSI, I'd be able to find some, but I make sure that I'm not incubating it more than 3 hours. So
method do you use, not the sperm preparation method? How do you prefer? You also do
just single density, yes.
Okay, nobody does simple wash.
Can I add one more thing?
Yes,
Yeah, please go ahead. I'll
no. I just want to say I do simple wash if I do teaser.
Okay.
So another thing that I had tried and it worked and gave me a live pregnancy also. That's why I'm discussing and like to share this is Zeta method. So you can do density and then followed by swim up. So you need a bit of like 0.5 ml at least of media in that. And then with just our rubber glove, okay, just rub the tube with that rubber glove. It actually creates a negative static. And because of that, whatever sperms are achieved at the tip will be all good DNA. It's actually a version of Max.
Come again.
So Zeta method.
It's very interesting. Yeah.
It's in WHO. I mean, it's more or less 30, 40 years old method. Nothing new actually.
Well, we still need a demo.
Yeah, next time you do, you record and share.
Yeah.
Record and share. Okay, is Dr. Zakaria, you would like to add some point in this? You are not audible.
Yeah. Unmute yourself.
Yeah, just concerning the technique of which we will proceed to prepare the sperm. I don't know if you have read the recent article, original article from the Journal of Fertility and Sterility, which is talking about the outcomes. The outcomes when we are using whether sperm preparation using density gradient and simple wash, and they found that the clinical outcomes are the same, even with using a simple wash or density gradient. So I just want to mention this, and it's a really interesting article. It must be read. Also, it dates of.
Yeah, I would like to ask one more question in this particular thing. So, how in case of simple wash method, the RBCs will be more. So, how do you manage that? And sometimes you get needle get blocked for juniors, so they can understand better. So, how do you prefer in those kind of cases?
I didn't understand the question.
In case of simple wash, usually when you make a ICSI dish, there will be lots of RBCs will be there. Right?
Yes, no, I'm just talking about the clinical outcomes, which means the live birth rates and the fertilization rates are the same. They found that they are the same between the simple wash and the density gradient. So it will avoid us to discuss the technique that we will be using in preparing our sperm, because the results are the same between the two techniques.
Kumar, you would like to add some?
Yeah, fine. See, now once you use the simple wash method, right? So then we will make like more MOPS media and we load entire sample. Then we incubate for like more than 3 hours or 2 hours so that the motile sperm start coming to the periphery of the droplet. So that we are not not disturbing any debris. We are only selecting the sperm which is come to the periphery after a certain time of the incubation.
But most
When
of
the simple wash.
Right?
Sorry?
Most of the sperm won't be motile in teaser sample.
See, now once it is a process with a simple wash,
Yeah.
after that we load all the sample to the ICSI dish with we load entire thing to the we'll make the MOPS droplet and line. We load everything. We incubate for the 3 hour, 2 hour. The motile sperms will come to the periphery. Then we pick that sperm and we'll put it in immobilization.
Nishad, you were saying something.
Yeah, just for the explanation, swim up as mentioned by Dr. Zakari, it's very true that swim up or DG gives you same result. So in case you're doing swim up also, you can use that rubber glove, rub it against your tube, and because of that negative charge, the sperms which are alive will move there very slowly.
You do
Just
them
pick,
on this.
just pick them up, the tip, keep it in a separate tube, and then you can use it for ICSI in the PVP.
So I would like to add some point in this especially. So we do a lot of micro TSA in our hospital, like 10 to 15 per month. So I think it's important to tell our surgeon to give less blood. That's the first thing. And second thing, whenever you get the tissue, I usually wash with multiple, before teasing, I usually wash multiple times. So I try to get rid of the most of the blood in that. And then I take the, I try to tease. Then I'll do simple wash. Before, I think this starts from when you get the tissue. I think that would help to reduce a little bit of RBCs in this case. Either you do a density or, but I follow simple wash method.
Shamshir, I just want to add one point. I also do the same procedure like you are doing. After multiple wash, if still you get RBCs in the pellet after centrifugation, leave it for some time. Take the sample above that pellet. You will definitely get sperms in the ICSI.
Yes, yes. So most of the time we know as far as juniors, so they can understand all this. So most of the time you won't see the motile sperms. So try to wash your tissue as much as possible to reduce the RBC, and make sure if every time if you get a tissue with the blood clot, please inform your surgeon first and tell. So you communicate, try to communicate with your andrologist and tell them we need
Yes.
less blood. So it starts from there, I think. So they will also, when you take the tissue and immediately wash it and put it in another dish, maybe you will get less blood, because whenever we get it, then the blood will get clot. It's very difficult to remove. So when you inform the surgeon, so he will wash once and maybe he will give in another dish. So this would help. One more point to be added. So any other method?
I just want to add one more point, Shamshir, that in this case, like these case, severe oats is getting very common nowadays. This is happening most of the time, that severe oats, and on the day of pickup, we don't get any sperm. So what we people are doing is we always keep 2 to 3 frozen sample as a backup. Or during the time of stimulation, like after 3 to 4 days, we take a sample and freeze them, so that we will not face this problem on the day of pickup, and though we don't want to go for TISA.
Okay. So next slide. Are we concluding this? Slides is not visible.
Yes. I think we are now end of this fantastic panel discussion. A lot has been
Rahul,
discussed
Dr.
about.
Zakaria wants to say something, I think.
So Zakaria would like to add final points.
Please unmute yourself. Yeah. Dr.
Zakaria, you can unmute yourself.
Yeah.
Do you hear me?
Yeah.
Please go ahead.
No, I didn't want to say something, but I didn't want. You can continue.
Okay.
So Rahul.
So yes, now we are finally came to end for this fantastic panel discussion. Thank you all the panelists for creating such a good atmosphere here and getting so much of insights related to sperm selection, getting so much of information, mostly for the juniors who are practicing and who have started for their in the embryology practices. So yes, wait sir.
Wait sir, please.
Yes, wait sir.
Yeah, I am here. I'm here.
Okay.
So thank you everyone. Thank you so much, all the panelists and moderator and all the speakers. It was wonderful, and I also learned so many things from you people and the new techniques what you are using. And I think one more thing I want to say, that Dr. Zakaria has joined us, and he's from Morocco. We were not able to introduce him properly, so I take opportunity to introduce him. He is from Morocco, and he is the HOD of the andrology lab in Morocco, one of the big center. And I met him on LinkedIn basically, and he regularly posting very good stuff on LinkedIn. So welcome, Dr. Zakaria. And now at the end, I would like to add Dr. Charu that Joshi already told you about our micro MBAT. So be ready for that one in-person meeting we are going to do on 8th and 9th June. It is all about that embryo culture. So we are discussing only embryo culture this time for 2 days. So we have 4 international faculty also, and Dr. Jason Swan and Dr. Nikati Findicali from Brussels, and Dr. Alejandro Bartiloki from Italy, and Dr. Thomas Abnar from Germany. So all four are coming, and with our national speakers. And our scientific committee members are also there, Dr. Nishat Chimote and Rahul, and few people are also there. So we are preparing the program, and daily we are sitting. And yesterday night we were discussing all the program, scientific program. So I think it will be very good, and you must learn from so many things from our scientific committee and our international and national speakers. So do join, and last date for early bird is 31st March. So it is very less fees, so anybody can join. And we are going to we are just trying to make some to workshop also. So it will be on media and disposable and TQM and instruments. So definitely you will get some good insight and update it yourself. So do join for that. Thank you so much, everyone, for joining us. Thank you so much. And thanks to Bharat Serum also for supporting this event. Thank you so much. Thank you.